Hello, my name is Stephen Vigil, February 7, 2020. This is week 4 lab, Bio 225, and today I'm going to be doing the experiment 1 for methyl red and bogus for testing. There's 2 parts on this, so I already got everything. I already done this part, but I'm going to kind of go over the motions, kind of what I did. So I already have all my supplies here. I already labeled my MRV Proth, Epi, and S. Epi, and E. coli. Those have already been labeled. So I'll kind of go over the motions what I've done. So I got my candle lit already. I'm just going to do the E. coli. So I have my broth from the E. coli. So I have my culture vial for E. coli. I will remove the cap. I'm going to run it through a few times. Sterilize the lid. Put the lid down. Remove the pipette out of the alcohol. Shake it dry completely. And then I'm going to pipette amounts of the E. coli. Run this through the sterilizer a few times. I'm going to put the cap back on. And then I'm going to get the MRVP of the broth. Open the lid. Run it through a few times. Sterilize the cap. And then I'm going to pipette four drops into the active broth culture. 4. Run this through the lid again. This goes in the undiluted bleach. The pipette goes in bleach. And put the cap on. Now I'm going to do this thing for this S. Epi, and I'm going to let this sit for 48 hours and let it end the incubation. And I'll pause this video so I can do part 2. This is part 2 of my video. So after I'll just continue from here. I had the broth that I incubated for 48 hours. So those have already been poured into 4 separate vials. So I had to create 4 vials. 4 vials here. 2 of them S. Epi and 2 of them E. coli. And I have already labeled them with the name on it. So I put half of the broth, half of the S. Epi in 2 of them, and half of the E. coli in 2 of them. Once I labeled them, put the broth in and everything, I'm going to get the methyl red, and I'm going to pipe it into the only 1 S. Epi and 1 E. coli. I'm going to put 7 drops in the E. coli and 7 drops in the S. Epi. Once I'm done with the methyl red, I'm going to dispose of that, put that in the trash. Clean the scissors with bleach. Once I'm done with that, I'll take pictures of those 2, which I have already done so, and I'll upload them in my lab. Part 3 of this would be to grab the tip of the Barrett's A regranite pipette. We're going to put 12 drops in each, so 12 in 2 of the E. colis and 12 in 2 of the S. Epis. And then we're going to gently mix them together and swirl them around. I have them standing up like this in the vial holder because the holes aren't big enough so they can stand up, so I've kind of had to improvise. Once that's done, I'm going to get the Barrett's B regranite, and I'm going to pipe it 4 drops in each, so 4 in the E. coli, in both E. colis, and 4 in the 2 S. Epis. I'm going to let that sit for about 30 minutes, and then once I'm done with 30 minutes, I'll be able to tell which one's in positive and negative. And as you can see, these are my end results. And these load pictures will be uploaded in the lab report. And this concludes my video. Thank you. For catalase testing, this test is the presence of catalase and enzymes that breaks down the harmful substances of the hydroperoxide into water and oxygen. If the organism can produce catalase, it will produce bubbles and oxygen when the hydrogen peroxide is added. Bubbles are present, it'd be positive for catalase, and if they're not present, it's negative for catalase. So I have my E. coli here, my E. coli agar plate here. So you notice it has several colonies on the back. I'm going to get my inoculation loop. I'm soaking in alcohol. I'm going to kind of clear it dry. Once it's dry, I'm going to open the agar plate, scrape across for any colonies. I have already labeled my plates placed beforehand with S-epi and E. coli with a dime-sized circle in the back. I'm going to smear the colonies onto the test slide. Once this is done, I can throw that in the trash. And then the hydrogen peroxide I have here, I'm going to pipe it. One drop of hydrogen peroxide to each area of the circle. Like I said before, it'll bubble if it's positive for Catalase, and it will not bubble for negative of Catalase. So in this case, I'll upload my pictures in the data file, but I had a positive result for an S-epi. And this concludes my video. Thank you.